Hello, this is Miss Evelyn again with the CNM Veterinary Technology program. I'm continuing in the microbiology part 3 PowerPoint. We left off with this slide, Streptococcus, which is sort of a bit of a review and also brings us on to the next slides. So streps can be broken apart into Landsfield groupings, and that's what we're going to do now. So again, if you were to outline this, you may want to start an outline section about Landsfield Group A and B. Now these are all various streps, okay? So a Group A strep, Streptococcus pyogenes, pus, purulent material. Streptococcus pyogenes, again, genus, species in italics. Again, we said that all of the groupings A, B, C, F, and G are beta hemolytic. So again, you can look at that hemolysis pattern and understand what that means. Okay, beta hemolytic. So group strep A's are beta hemolytic. Your cell wall antigen complex is a polysaccharide. And some of these group A streps may have a capsule that's composed of hyaluronic acid. That sounds like something you might have inside your knee capsule at times, but it's a capsule composed of hyaluronic acid that protects them, makes them work in different ways. And there's an M protein, a cell surface antigen that helps resist phagocytosis. Phagocytosis. It helps it resist phagocytosis, which is one of the reasons that strep pyogenes is sometimes harder to get rid of, because your white blood cells cannot maybe come in and phagocytize it as well. So group A, Streptococcus pyogenes, they are beta hemolytic, and they have issues with their cell that gives them different structures. Streptococcus agalactiae, say that with me, Streptococcus agalactiae, group B strep, again beta hemolytic, quite often with mastitis. Now you noticed, if you remember, some of the staph also can cause mastitis. But here we have a strep that might be associated with mastitis. Landsfield groups C and D. Okay, so again, the C is beta hemolytic. Group D is non-beta hemolytic. Okay, so that's important to remember. There's Landsfield group C has a carbohydrate on the cell wall. It produces hemolysins, and some animal infections are most noticeably in the horse is strep equi and strep equisimilis. So doesn't equi make you think of equine for horses? So strep equine and strep equisimilis. So remember, group C streps, they are beta hemolytic, and horses. So again, kind of follow some of the underlinings that I have here for some of the items to try to remember about these various organisms. You are not going to see all of these in your hands-on labs. Most of the time, any streps that we have, we may not get down to a species name. And again, it will say it is a probable or potential or suggestive of strep. Group D strep. So this is not beta hemolytic. It's not what we would call an enterococcus, and it's a harmless inhabitant of the animal intestinal tract. But you could also, the Streptococcus bovis. Doesn't that think about bovine? So it is harmless if it's in the intestinal tract. But if it gets out of the intestine, you had to do surgery, and you had to make an incision into the intestinal tract, and your surgeon was and yourself were not prudent with your care, that strep, if it got someplace else in the animal, may be a big problem. But harmless inhabitant of the animal intestinal tract. Okay, so you notice a lot of these streps seem to have animal names related to them. Streptococcus suis. So suis should make you think automatically about pigs. So Latin terminology for pigs, basically. Streptococcus suis. It's an important zoonotic pathogen, so it can transfer from animals to humans. It can cause meningitis. This is actually a Lancefield group R and S. Sepsis, and a lot of times arthritis in young pigs. Okay, most cases of human infection come from people in very close contact with pigs. And again, in some third world countries, humans and their animals live much differently than we do in the United States. And those pigs might be roaming the house. They may sleep close to them. All different kinds of things that, you know, and that human infection can come from the people in very close contact with pigs. If you just pet a pig, more than likely you wouldn't get it. But if you were doing veterinary medicine to a whole herd of swine and there was Streptococcus suis there, could you get it? Possibly, especially if you had some kind of a cut or an abrasion. So it is a zoonotic pathogen. Streptococcus viridans, viridans, V-I-R-I-D-A-N-S. This is an alpha hemolytic strep, alpha or non-hemolytic. So it doesn't really do much to that blood auger. And many species of Streptococcus viridans can inhabit our oral cavity. And it tends to be an emerging as a significant pathogen because it is there in an oral cavity. So again, it is important when you are obtaining an organism in the microbiology lab, or if you are sending a sample to a lab, that you're very specific about the species that came from. Did it come from pigs? Did it come from a bovine? Did it come from an equine? Did it come from the oral cavity? Because all of those are clues for somebody in a diagnostic lab to maybe think which organism might be the highest probability. Streptococcus pneumoniae, abbreviated SPN, Streptococcus pneumoniae. Okay, pneumoniae is the species name. This is a gram-positive cocci in pairs, and they call it lancet shape. I will try to draw that a little bit here with my pointer. Okay. By lancet shape, it would actually be kind of drawn out, and then it would have a point, just like a lancet. Like you have a lancet, so straight, up to a point, and a point, and again, straight edge. So Streptococcus pneumoniae might be what they call lancet shape, and that can help. It is still a cocci, but it's not as round as some, and it's not oval, and they tend to have sort of a little point. So it would be also alpha hemolytic. It is, if you were doing a soluble test, it is biosoluble. There is an optogen sensitive or P-disc test, and it would be positive for that. The virulence factors, because it has a polysaccharide capsule around it, and there are about 80 antigenetically distinct capture types recognized, or more by the time this is actually presented to you. And respiratory diseases. So again, where did the sample come from that you're sending to the lab? Is it from the oral cavity? Is it from a wound? Or is it from a deeper respiratory area or sputum or something on that order? And that would, again, could help a diagnostic lab have a better idea what they may be looking at. And they may be more cautious about how does the hemolysis pattern look. Can we do a bile test and actually be able to see if it's soluble or not? Major concerns surrounding our SPNs is emerging strains that are resistant to penicillin. And it has altered our penicillin binding proteins, decreased affinity for our penicillins. So Streptococcus pneumoniae, know a couple of those main parts. We now kind of shift over to Enterococcus species. Entero, so more so into the GI tract, the entero, the small or large intestines, and again, Enterococcus species. These were at one time classified with group D strep. Classification of microorganisms keep changing all the time as more and more tests are developed and testings are done, and it may shift with what group they're in, but it just helps you to understand that it may be a little bit of a floating division at times. Again, normal residents of the gastrointestinal tract, but if they get out of the gastrointestinal tract into a wound, into the abdomen during surgery, then you may have a very bad problem. Increasingly resistant to antimicrobial agents, to your antibiotics. It is already resistant to penicillin, cephalosporins, and aminoglycosides, and also vancomycin. So this is the problem that we have, is that Enterococcus, if they get out of their natural flora area of the gastrointestinal tract, our antibiotics to be able to go after that infection may begin to be limited. These also like to grow definitely at a 10 to 45 degrees centigrade. They may like a little higher sodium chloride, pH of about 9.6, and they produce PYR. So a lot of these would be tests, the esquilin hydrolysis, esquilin and producing PYR tests, probably more so only in a bigger diagnostic lab. Okay, so a couple of these, and remember that these are in the normal gastrointestinal, especially lower gastrointestinal tract. So they kind of sound like fecal. So Enterococcus faecalis, F-A-E-C-A-L-I-S. Enterococcus faecalis is the species, and Enterococcus faecium, F-A-E-C-I-U-M. So Enterococcus faecalis and Enterococcus faecium. To me, that kind of makes me think about fecal matter. Again, normal in a GI place, but if it gets out of the GI system, let's say that that organism is present and a animal comes around, gets next to the feces, has a wound, and the wound is contaminated by the feces, then you may end up with a very complicated issue. These used to be with Lancefield D groupings. They can cause complicated urinary tract. So again, remember that these couple of the little points, Enterococcus faecalis and Enterococcus faecium would be normal in the lower GI tract, but if you had a diarrhea situation or feces that contaminated to the outside of the vulval area or the penile area, and the bacteria transmitted into the urinary tract area, then you can have a very complicated urinary tract infection. Bacteremias, endocarditis, intra-abdominal pelvic infections, wound, soft tissue infections, neonatal sepsis. So again, if the mother had this and the little neonates got up next to the feces that was contaminated, they might get a very bad infection around their umbilicus area and so forth. So Enterococcus faecalis and Enterococcus faecium, normal in the GI tract, get someplace else, can have big problems, and because it's normal in the GI tract, it might infect into your urinary tract, vulvar or penile areas, and urinary tract infection. Aerococcus species, Again, genus is capitalized, italicized, species in this case. Many different species might be here, so it's just species, SPP, in small case. These are strep-like organisms. They're found in the environment, and they're opportunistic. All right, so now we're changing to what we call the veterinary technology, or vet tech enteric pathogens. Again, enteric, we're talking about what is in the GI area, okay? So most frequently encountered bacterial isolates are recovered from clinical species. That's what we're going to be looking at. These are actually found in the soil, water, plants, and in intestinal tracts of animals and humans. Again, in your laboratory setting, if you were to see one of these, you could use the word presumptive identification or possible, suggestive of. You are not saying that, no, for sure I have something, but it is presumptive, suggestive of, or possible, those kind of words. It is on the gram stain, it is a short, plump rod or coccobacilli. So rod shape, or sort of maybe a little bit more oval shaped. Cocco, sort of round, but sort of oblong as a rod, okay? And they're gram negative. So what color is that? You should know that immediately. That gram negative is in the pinkish reddish area. On a blood agar plate, the abbreviation for blood agar plate, BAP, they tend to be very large gray colonies, sort of dry or mucoid looking, and their hemolysis on that blood agar plate, BAP, you should know that abbreviation, is sort of variable. It might be alpha, it might be beta. And quite often these organisms, these enteric pathogens, have what we call thin films or waves over the agar, like a swarming Proteus. Really, we talk about it as waves, like you're on an ocean beach and you have the tide coming in and you see the waves kind of formation on the sand. That's what this looks like. I believe I'm going to pause for one moment. I'm going to switch over to another document. I'm not pausing, I'm just stopping my sharing. And let me find my other document that I had pulled up. This is a reporting document. And I think I can increase this for you a little bit, and I will use the sidebar. So this is a suggested layout for when you do your reporting in a hands-on lab. Information that you would need to know. Where did it come from? Or your source. If it is a known, then you would write down, I have a sample of Staph aureus, and you would be trying to get your presumptive diagnostic work done accordingly. But if it's an unknown, okay, it is a mixed colony, or it came from a swab of the mouth, came from a wound, where is that source? Is it a known organism? Is there a source? And the species should be included. Did it come from a canine, a porcine, an equine, oral cavity, wound, where? All of that helps you understand some possible presumptive guesses to that organism. Then as you have grown that organism, and looking back onto your chapter, your unit 7, and you'll see the diagrams about how does the look of the organism look. The colonies, first off, you look at your plate that you have, and you look how the growth, your observations, your growth examinations, you look at that, and you say, is it a smooth colony? Is there rough edges around the colony? Is it wavy and dry? What does the surface look like? You're going to make notes about that. And is there a color? Is it yellow? Is it red? What auger did it grow on, and what color is it? That can be different. We will illustrate to you how to do what we call wafting, W-A-F-T-I-N-G, wafting, to see if there's an odor. You don't just put your nose into the Petri dish, you wave your hand, you waft it across that Petri dish area and see if there is a fruity odor. Does it smell like popcorn? Does it smell like dirty socks? And you can use those kind of terminologies because that's how we kind of list them at times. And then in your gram stain, what is the form and the shape of the colonies there? And include your observation and make drawings. You're going to do a exercise this week where you're going to actually almost utilize this kind of form layout. Be thinking about how you would actually illustrate a certain colony if you saw it on a microscope. Again, what's the margins of those colonies? And what media does it grow on? Gram stain, is it gram positive, is it gram negative? And the morphology, is it a cocci? Is it a cluster of grapes? Is it a chain of cocci? Is it lancet shape? Is it a rod? Is it gram negative and a rod? Okay. And then what tests you may have done. And that may even just include a hemolysis test. If you grew it on BAP, blood agar plate, and you have a hemolysis pattern, what is it? You may not have done any other tests than knowing that. So at the bottom part of this form is giving a sample, okay, of how that might be. So strep pyogenes, the source might be the computer keyboard, because you will do a exercise where we have you do sort of a hunt. You get to go and find something in and around campus, your personal belongings or yourself, and try to grow that out on a plate when we have our hands on lab time, sort of a scavenger hunt. But slightly raised colonies, they're white. There's no, so again, you do not just leave a line blank on a reporting form. If you wafted, W-A-F-T-E-D, if you wafted across that plate and there was no odor, you write none detected. You don't leave the line blank. A blank means, did you try to do that or not? We have no idea. So if you say none detected, we know you tried to find that. Okay. What form? Punctiform to circular. And you can see some of the discussions in your textbook on how those are actually terminology you can use. The even edges around the colony. Grows on what media? Blood agar had beta hemolysis present. It's gram positive. It's cocci in clusters. It was catalase negative. Okay? Because it's a strep, and you ran a catalase test with hydrogen peroxide. It didn't bubble. It was negative. So that is ways that you would actually utilize this kind of a form, and this is in your documents in your Blackboard shell. You can find this form. I recommend that you download it and have it ready for next term when we have the hands-on lab, and be ready to utilize this. And again, for unknowns, always include that terminology of presumed or potential or suggestive of. So without being in a diagnostic laboratory, you really do not know what that organism is. You are making a presumptive or potential educated guess. If you call it strep pyogenes and you're not in that diagnostic lab and you don't include the word suggestive of or presumed or potential, you would not get full credit on your answer. Okay? I think I'm going to pause this again at this moment and break it up into another recording session.